DNA stores the genetic information needed for cells to function. Before a cell divides, this DNA must be copied so that each daughter cell receives a complete set of genetic instructions. This process is called DNA replication. In eukaryotic cells, DNA replication takes place inside the nucleus. Prokaryotic cells lack a nucleus, so they replicate their DNA within the nucleoid region of the cytoplasm before cell division. DNA replication begins at specific DNA sequences called origins of replication. At an origin of replication, proteins assemble and recruit the enzyme DNA helicase, which begins opening the DNA double helix. This creates a replication bubble with a Y-shaped replication fork at each end. DNA helicase then moves along the DNA, breaking the hydrogen bonds between complementary bases and separating the two strands. As helicase unwinds the double helix, the DNA ahead of the fork becomes increasingly twisted. Another enzyme, called topoisomerase, relieves this torsional stress, preventing excessive supercoiling. The separated DNA strands naturally tend to rejoin or fold back on themselves. To prevent this, single-strand binding proteins, or SSB proteins, attach to the exposed strands and keep them stable. The two parental strands can now serve as templates for new DNA synthesis. However, DNA polymerase cannot begin building a strand from nothing. It requires an existing free 3' hydroxyl group. To provide this starting point, DNA primase synthesizes a short RNA primer. DNA polymerase then attaches to the primer and begins adding complementary DNA nucleotides. Adenine pairs with thymine, while guanine pairs with cytosine. In this way, the sequence of each parental strand determines the sequence of its newly synthesized partner. DNA polymerase can extend a new strand only in the 5' to 3' direction. Because the two parental strands are antiparallel, the two new strands must be synthesized differently. The leading strand is synthesized continuously in the same general direction as the moving replication fork. The lagging strand is synthesized discontinuously in short sections called Okazaki fragments. Each Okazaki fragment begins with a separate RNA primer. Finally, DNA ligase seals the remaining nicks between neighboring fragments, creating one continuous strand. Proteins such as the sliding clamp help DNA polymerase remain attached to the template, allowing it to copy long stretches of DNA efficiently. DNA polymerase also proofreads the newly synthesized DNA and removes many incorrectly paired nucleotides. Additional repair systems correct most of the errors that remain, making DNA replication extremely accurate. Replication continues until the entire DNA molecule has been copied. The result is two nearly identical DNA molecules. Each contains one original parental strand and one newly synthesized strand, which is why DNA replication is described as semiconservative. The overall process can be divided into 3 stages: initiation, when the replication machinery assembles and opens the DNA; elongation, when new strands are synthesized; and termination, when replication is completed and the newly formed DNA molecules separate. Although the basic principles are shared by all organisms, important differences exist between prokaryotic and eukaryotic replication. Most bacterial chromosomes begin replication from a single origin, while eukaryotic chromosomes use many origins simultaneously. Eukaryotic cells must also solve the problem of copying the ends of their linear chromosomes using structures called telomeres and an enzyme called telomerase.